of the Brotman Beatty Institute for Precision Medicine and a Howard Hughes Medical Institute investigator. Jay actually joined the UW faculty straight out of his MD PhD at Harvard. Since then, he's been recognized as a pioneer in developing and applying new genomic technologies. So his many awards include Young Investigator Award from the MIT Technology Review, as well as the Kurt Stern Award from the American Society for Human Genetics, and an NIH Director's Pioneer Award. And so today he'll be talking to us about quite ambitious genomic technology application, reconstructing prenatal development. So welcome, Jay.
Thanks, Kelly. Great to be here and have a chance to give this talk. So it's always a little tricky to know how to kind of pitch or the right level to hit with a more of a public-facing talk where you don't really know what the level everyone's level might be, right? And so I'm going to try my best. Unfortunately, I've got a friend here, Chris, who I've known for a very long time, and so I know what he knows and what he doesn't know. And so it's like a great level to just kind of like, okay, I'm going to finally, after all these years, tell Chris what I do. So, okay. So at a high level, I just thought would start with this kind of nice little point, which is that trees are a recurring theme in biology, right? So the image on the left is from Darwin's notebooks, right? And thinking about trees as species and how species are related. The middle of course, is a pedigree, right? Showing relationships of individuals in a family. It's a different kind of tree. And on the right is a tree I think we think about a little bit less, which is a tree of cells, right? And that's primarily what I'll be talking about today, right? And so what I mean by a tree of cells is referring to the fact that, you're in a department of genome sciences, right? And so you think about what a genome does. The genome is essentially encoding a program of exactly this, of a tree of cells in a developing organism, right? And basically what that means is that you're starting, we all started from a single cell, right? And that single cell divided and divided to give rise eventually to a child, and then, the child will continue to grow, right? But even just in these, 9+ months, we're talking about a single cell with a single genome, dividing and dividing ultimately to give rise to roughly, trillions of cells, but in a very well-organized and functional form, right? And this is true, this child here, which happens to be one of my own, it's also true of, each of us, right? So this process of 1, 2, 4, 8, 16, dividing and dividing, eventually giving rise to about 40 trillion cells in an adult human. We're not uniform, right? Meaning that we're composed of different kinds of cells. And so you can imagine that, somewhere in your stomach, there is a cell that traces, every cell traces its lineage back to this single cell zygote, but it does so through a certain path. And other cells, say in your brain, trace their heritage back through kind of a different story, right? And so those stories are all kind of embedded in our personal histories of development, right? So what does a genome have to do with any of this, right? So the genome you can think about essentially as a recipe book for building, for encoding this program, right? So the actual genome is just, like a string of letters, A's, G's, C's, and T's. 3 billion of these in a row is kind of what you inherit from one parent, another 3 billion from another parent. And the way I would describe this is kind of like a recipe book, right? And the recipe book has some degree of structure, right? Ultimately, let's say your, one page might encode for cookies, right? In the genome, we have genes, right? And those encode for proteins, right? So the genes are the recipes, and the proteins are essentially the cookies or other kinds of food, right? And the genome altogether has around 20,000 recipes, right? And when we think about, in every cell in your body, there's a chef, right? And that chef proliferates and is present in the daughter cells of that first cell. And but the chefs will change over time, right? And so they might be interested in making different kinds of things. So you don't end up, turning into a homogeneous blob of all cookies, so to speak, but rather over time, different cell types will start making different sets of recipes, different meals, so to speak. And that's why, essentially what makes a liver cell different from a liver, from a brain cell, or from some other cell, right? They all have the same genome, but they're essentially choosing to make different subsets of recipes, kind of indicated by, you know, the bookmarks that you might stick into different pages, let's say in a brain cell versus a liver cell, that might give rise to different kinds of things. So one recipe book, your recipe book or my recipe book, just used in very different ways, right? And so if you think about what we are, right, we're essentially a giant blob of organized cells, right? And that program, again, encoded by the genome, also encodes the genome of, let's say, a fish, right? So a different DNA sequence, also starting from a single cell, but giving rise to a very different form and function by virtue of the same kind of a tree, right? It's playing out over, let's say, weeks rather than months, depending on the kind of fish. And this is true, essentially throughout nature, right? So the phrase that Darwin used to describe it was endless swarms most beautiful, right? Referring to just the incredible diversity in both the plant and animal kingdom, but really all of which kind of starting from the same basic motif of a single cell dividing and dividing to give rise to all kinds of wondrous differences in form and function. And yet, at the same time, as we think about all these trees, they're clearly very different, and yet they have a lot in common, right? And so this is one of my favorite examples. This is something I came across as an undergraduate, and this is actually one of kind of the things that got me hooked on genetics in particular. And the trait here is called the piebald trait, and you can see that kind of white forehead blaze on the kid. And it's caused by a mutation, a deletion in a particular gene called c-KIT that consistently and reliably results in this trait when you have it. And then if you look at a mouse where you delete essentially the same gene, right? So we're related to mice, and so there's a gene. There's also a c-KIT gene in the mice. You get this trait that looks almost identical in terms of the physical distribution of where these white blazes are on the body, right? And I think this kind of makes the point, I think that, on the one hand, there are incredible differences across, let's say, the animal kingdom or the kingdom of, mammals, let's say, and yet there are these deep similarities that persist, and it can sometimes be revealed by, the genes that underlie particular traits that might look very similar in humans and mice, right? I also love this image because it kind of just going back to one of the earlier slides, that kind of encapsulates kind of what all three kinds of trees in one image, right? So on one hand, we're looking at a small subset of the tree of species, right? Mice versus humans. We're looking at essentially trees of inheritance in the sense that this is a genetically inherited trait that you would see passed down in families with particular patterns. And then finally, we're seeing this deep homology between across 60, 70 million years of evolution in terms of how deletion of a particular gene manifests in terms of what you see in the organism that results, whether it's a mouse or a human. Okay, so this is part of the reason for why we want to study development, right? So we want to understand human disease, and we can do that by studying the development for many diseases, and we can do it by studying model organisms like the mouse and other models because they are relevant to human diseases, as this example shows, right? So, okay, probably the foremost example of really a crazy achievement in this field is by this guy over here, the late John Sulston, who was a British scientist who very famously sat in front of a microscope for 2 years, morning till night, and watched the development of a worm, right? So every day he would get up, go sit on the chair, and just look through the microscope and watch these worms developing. This is a roundworm called C. elegans, which we study, still study a lot. And the cool thing about C. elegans, this worm, is it's very small. So as an adult, it's only around 1,000 cells. And it's also transparent, so you can watch the whole thing, right? So it's not like he watched one worm developing for 2 years. Rather, he would watch thousands of worms developing, and he would write down in his notebook what he saw happen and then try to reconstruct the whole thing, right? And in a few slides, I'll kind of go through that in a little more detail, what I mean by that. And so what we'd like to do, this was 40 years ago that Sulston did that, right? And we still haven't done anything remotely approaching that for any other organism, let alone something as complicated as a mouse, which is far more complex in terms of its development compared to a worm. And the reasons are kind of obvious, right? So for one, you can't see through a mouse, right? This worm develops exactly the same way every time, always exactly the same way. And so it made it made it possible for John Sulston to kind of piece together what you would see in different worms, whereas even identical twins, for example, will develop slightly different ways, and the same good thing goes for identical twin mice. And then finally, there's a question of scale. The worm, as I said, was only about 1,000 cells. In a mouse, we have orders of magnitude more cells, right? So just to come back a little bit to why we want to do this and kind of why the mouse in particular, right? So about 20 odd years ago now was the sequencing of the human, maybe 25 years, no, 20 years ago, the human genome, very quickly followed by the mouse genome, both of these projects led by people who are still in our department. And this is the program, right? This is like the code, right? But in terms of what it actually codes, it's the output of that program is the phenotype, right? What we call the phenotype as opposed to the genome, which is the genotype. And so we still don't really have a good handle on this for either the human or the mouse, right? Or really for any other organism other than the worm, right? The nice thing about, a few nice things about the mouse in particular, right? So kind of going backwards here, one, it's accessible. We can't study human development in the same way for obvious ethical reasons. It's very reproducible, right? So we have mice here in the building, people have mice in Australia, people have mice in England, everyone has access to the same model, and it kind of happens the same way every time. And it's also bounded, right? Like we can hold a mouse, right? We physically know what the extent of it is. And so, it's not like a limitless thing. And so it's in principle something that's solvable. And then finally, I made this one example here, but there are endless other examples. It's an amazing model for human biology, right? And much of what we know about human biology is actually based on the mouse. So this is kind of why we're doing it, right? So the next question is how to do it, right? And so the first thing we could contemplate is something like what Sulston did, which was just to watch, right? And just to give a little more granularity to what Sulston actually did, right? So on the right here is the final, or I guess I shouldn't say the final tree. It's a subset of the tree that Sulston obtained. And just to give you a sense of how he was doing it. So on the left here, actually images from his original notebook, where he's actually drawing cells and kind of saying this cell goes to this cell. And then here's kind of a more, formal version of that. And he basically just pieced through, right? He's just watching little things and converting his notebook drawings into a tree, right? And in these trees, basically you've got an origin, right? And that cell will divide into 2 branches, and those branches will divide into 2 more branches, and so on and so forth, kind of proliferating out. And he would name these cells things like A, B, A, right? Where every cell had an A daughter and a B daughter. And so if you named a cell A, B, A, you knew exactly what cell you were talking about. And so he went on and did this more and more and so on and so forth, right? Okay, again, kind of to come back to the point from earlier, you can do this because you can see through the worm, right? If you wanted to, do the same thing for the parrot circulatory system, you couldn't do that, right? Because you can't see through a parrot, right? Or the eye of a wasp or the brain of a mouse, right? There's endless examples of amazing, beautiful biology that is simply not visible to the naked eye, right? Or to a microscope or really anything, other technology that we have right now. So we're at, this is why it hasn't been done for 40 years, right? Okay, so do we have other options, right? So most of what I'll talk about today is we have 2 other options, and I'll come back to the third option at the end. But I'll mostly talk today about the second option, which I'll call sampling, okay? And so the basic idea here is that we're not gonna study one mouse, right? We're gonna study lots of mice, and we're gonna try to catch them at different moments in time. And we're gonna, if we can catch just enough of them, so many of them, we can kind of potentially piece together what's happening by comparing this moment to the next moment and so on and so forth, right? So kind of a sampling and stitching type approach, right? And so to do this, we've been relying a lot on a new generation of technologies that we call single cell profiling technologies. And I'm going to gloss over a lot here just for the reasons of time, but our group, together with other groups in the department, another lab called Trapnell, have been spending a lot of time trying to profile entire organisms, so like an entire fish or an entire tadpole or an entire worm with this kind of single cell resolution, right? So not focusing on a particular organ, but trying to get the entire thing, right? And again, without getting into details, part of the reason we've been able to go after this has been investment in technology, and in particular, technology for profiling very large numbers of single cells, right? And so there are methods actually developed here in the building, in particular, former postdoc Darren Kasanovich and still a current research scientist here, Riza Daza, developed a method that allows us to profile. I'll just summarize it as profile cells in an exponentially scaling way. That means if we do twice as much work, we can get 4 times as many cells out. If we do 3 times as much work, we can get 9 times. So like it basically goes up and up at a much faster rate than linear scaling, right? And so by virtue of this, just 2 labs have been able to profile a very, very large number of cells in a relatively short order of time. And this also, in the wake of kind of developing these technologies, it was kind of like you have a very large hammer, and now you're going around looking for something to hit with it, right? And this has actually got us first interested in the mouse because from a complexity perspective, the mouse is a pretty big target for a big hammer, right? And so these beautiful images I'm showing here are from a window in mouse development that's called organogenesis. And so mice develop prenatally over about 3 weeks, right? Which is not to say, so a mouse, from the fertilization of the egg to the moment of birth is about 3 weeks, right? And that's not to say that a 3-week mouse is the equivalent of a newborn human baby. Rather, mice are born at a stage that are kind of earlier in development than, let's say, humans are. Humans stay in the womb till they're further developed, right? But you get a sense, that this nomenclature of, when I say E 9.5, that means that 9.5 days have passed since fertilization, right? When I say E 13.5, it means, almost 2 weeks have passed since fertilization. And you can see this embryo growing very quickly, right? Just over 4 days here, that increase in size is remarkable, right? And so, again, without getting into details, former trainees from the lab, Jinyu Kao and Malte Spielman, did an experiment a couple of years ago where they basically took embryos from this stage of development, and it's called organogenesis because this is when the foundations of many of the organs in the body are being first formed, right? So the initial laying out of a pancreas or a stomach or what have you. And you can kind of see some of that in the images here if you squint. And so what they did was to profile embryos from each of these stages at a scale that really no one had ever done before, right? So they were able to profile 2 million cells in one experiment, which at the time was the largest experiment of this kind that anyone had done. And when I say profile a cell, what they're essentially doing is asking what's inside, right? In terms of what recipes are actually being made in any particular cell, right? But they're doing that for each of 2 million cells, right? And so you might say, hey, 2 million is a lot. That sounds great, right? But the truth is that it's actually only a tiny fraction of the cells that are in a mouse embryo. So I don't think we really knew the answer to this until pretty recently. So Beth Martin and my group will talk about a little more, well, the work I'll talk about a little more later is did an experiment maybe a year ago where she actually tried to ask how many cells are in a mouse embryo, right? And without getting too much into the details here, what she found is that a mouse embryo has about 8.5 days after fertilization, it already has 200,000 cells, right? At 3 weeks after fertilization, when it's born, it has 670 million cells, roughly, right? So it's growing, it's doubling at an incredibly fast rate. At this early time point, it's doubling every 6 hours in number of cells. So the whole thing is doubling every 6 hours. And that slows down over time. But even at the time that it's born, we estimate that it's doubling in size about every day and a half, right? Or day or so. Okay, so for people who are more have more of a computational bent in the audience, you might be wondering kind of what does data from these kinds of experiments actually look like, right? So I'll just briefly kind of show you that to give a sense of scale. So essentially, for each of these 2 million cells, we're asking what recipes are on, right? But our ability to do that is actually not that great, right? So it's kind of like we peer inside every cell and we get a very quick glimpse at what's going on, right? And we peer in another cell, we get a glimpse of what's going on. And so what we end up with essentially is a matrix, right? Like, you could imagine this in a very, very, very large Excel spreadsheet, right? Where you had 2 million columns, every column being one cell. And then you had 20,000 rows, every row being one gene, right? And ideally, the data would look like this, where we could say, oh, you're making 3 pancakes and 2 eggs and 13 pieces of bacon or whatever. Sorry, 3 pancakes and 2 eggs and 1 piece of bacon, right? Because these are the genes in this cell and kind of different numbers of those things in another cell. But because the technologies simply aren't good enough yet, it's mostly zeros and ones, right? So again, we have kind of this very crappy snapshot of what's going on in each cell, and we've got to kind of make sense of that, right? And so you can imagine that, dealing with, you can also think about this like a 20,000 dimensional object, right? So where if we only had 3 genes, we could plot it we could plot it in Excel, right? We could have like a 3-dimensional plot and look at the expression of each of those 3 genes in a very simple plot, right? But we can't do that because we have 20,000 genes and the human brain can't get itself around that kind of space, right? So we use various algorithms to transform this data structure into something that we can get our head around. So it's kind of a fun animation that Cole Trapnell, my colleague, made of this data set where essentially every point on this, so we've essentially taken this 20,000 dimensional object and convert it to a 3-dimensional object, right? In a way such that points that are near each other are more similar to each other. Every point here is a cell, and there are 2 million points represented. And when you see a big glob of cells, it implies that those cells are related in some way, right? And so what we can then do after the fact is we can look at, oh, I've got a big mass here. What do I think it is? And kind of based on known biology, we can find a small cluster that corresponds to the lens of the eye, other clusters that correspond to something we call neural crest. All of blood cells are in one place. All of blood vessels are in another place. Skin cells, the liver, and then various other things. And then this giant thing at the bottom is essentially all cell types of the brain, right? And then obviously there's a lot more here than just these 10 things, and we can take each of these things and break them down further. So for example, if we take the epithelium, or kind of the skin and things that kind of derive from a similar source in development, we see all sorts of different, I'll call these trajectories. So you actually have skin, but you also have the kidney, essentially cell types in the kidney and the lung and the gut, so on and so forth. And a cool thing here is that you can see the dimension of time here, right? So even though we sampled these cells from embryos at different ages, we can in fact see things stitching together in a way that fits with the progression of time, like a film, right? Where, if we look at the 9.5 moment in the film, the next thing we see, the next development is at 10.5, 11.5, 12.5, 13.5, right? So even though we don't necessarily, imagine we just took a bunch of snapshots and we didn't know what order they went in, simply by looking at the pictures, we can order them, right? And the order makes sense based on where we know we took the snapshots, right? Okay, so now you can imagine that just like in movies, right? The quality of a film depends on the frame rate, right? So if you have a crappy frame rate, you're gonna have a crappy film, right? You got a good frame rate, right? You can piece together a nice movie, right? Like one of the original films shown here, right? And so, at the time I think we did this experiment, we thought, it was amazing, right? Because we had 5 days, everything fit together. But we also realized, I think, how rapidly things were changing, where maybe even catching things every day wasn't enough because the embryo, as I said, is changing so quickly. So what we've spent a lot of time in the last year or so doing, so here I'm showing, this is like the 5 days, right? So 9.5, 10.5, 11.5, 12.5, 13.5. Essentially what we've been trying to do is trying to extend and fill in, right? So can we now capture at a much higher frame rate? Can we also capture earlier moments in development? And can we extend this all the way out to birth, right? To the birth of a newborn mammal, essentially, or pup, right? And so that's this was a very long introduction. I'm going to get to the meat of what I'm going to talk about. It's kind of this experiment and a little bit about what we learned. And so, I think this is arguably a big project to try and tackle something like this. And I think in science, you often run into things like this that are, massive team science, right? With huge armies and subcommittees and things like that. This was overwhelming the work of just 3 people, right? Did almost all of this, right? So I just highlight them here and I'll kind of go through the pieces that they each did. So this work was a collaboration with a place in Bar Harbor, Maine called Jackson Labs. And this is one of the foremost places for mouse genetics in the country. Has been for 60, 70 years or something like that. And so they were very kind, and this person, Ian Welsh in particular, in getting excited about this project and trying to help us identify embryos that kind of corresponded to that very fast film rate or frame rate. And so this involved precisely staging nearly, or I guess over 500 embryos, and basically looking at the embryos and trying to make a guess about where they were in development, right? And so for very early development, we think we have a frame rate about every 2 hours. And then it's a little bit worse as things go further along. So for the rest of development, about 6 hours, right? But still much better than we had, and still kind of much more comprehensive. So an enormous amount of work here, kind of condensed to a single slide, and just kind of highlighting some of the things that we look at under a microscope, or Ian looked at, to make a guess about what time point each embryo corresponded to. And then this next part, so those embryos were frozen in Maine and then shipped to Seattle. And then Beth Martin, who's a research scientist in the lab, did this next part. And it's hard to condense, I think, an incredibly large amount of work in terms of trying to improve the underlying methods and then apply them in a few slides, but I'm going to try to do this. I borrowed one slide from Beth, which is this one. And this is, I think, probably a great summation of the amount of optimization she had to do to get this protocol working well. And so it turned out that for years, as we were doing our experiments, our labs were like heavily contaminated with RNases that were destroying essentially the things that we were looking at, right? And so finding effective ways to stop those enzymes from screwing up our experiments, or at least reducing the quality of our data, was a lot of detective work and a lot of hard work to kind of get there. But once she got there, right, she then set about generating the data set, and essentially over 15 or so experiments conducted over a few months, was able to crank out 12 million of these single cell profiles, where we're essentially looking at the contents of each of now not 2 million cells, but the 12 million cells. And so I think it's good, I think especially if you're not in this field, you don't have a lot of context for, what 12 billion might mean. So just to kind of put this, I think it's a little bit of context, right? So, the Human Cell Atlas, which is an international consortium with, 600 labs and 400 projects, almost all relying on a single technology, has generated about 48 million cells worth of data, right? And so we have less than that, right? But this is one person, right, in one lab, right, with a different technology. And I think the person is different, right? This is a special person, I want to be clear. But a different aspect about this is also the underlying fundamental nature of the technology. Some technologies scale linearly, other technologies scale exponentially, right? And just taking advantage of that exponential scaling is what, a part of why we can operate at this scale with a very small team, right? Okay, that was a lot of work reduced to 3 slides. And then the analysis of the data has overwhelmingly been done by Chengsheng Qiu, who's a graduate student in the lab. And again, I'm not going to do justice to this in the short talk, but I'm going to try and highlight some fun, at least one fun story. So again, to kind of put a lot of work, like years of work during the pandemic into a very short thing. One of the hardest challenges here is annotating, essentially we're trying to annotate every cell type throughout the entirety of mammalian fetal development, right? And so, there's some cell types that we expect kind of what to see and we know what they look like, but there's tons of cell types where, we might expect to see them there, but we don't know exactly what they'll look like in our data. So this was years of work going back and forth and just kind of trying to piece through individual observations and figure out what we were looking at, right? And so here, for example, we could figure out here are epithelial cells and we can blow those up and okay, we think these cells are from the kidney, and then we can blow those up and find different parts of a nephron in the kidney in the underlying data, right? And so overall ended up annotating hundreds of cell types throughout the data. So not going to go through kind of every piece of what we found, but I'll just highlight a few aspects to give you a feeling for how we think about or how we look at the data, or the kinds of things we can see. So one fun thing to look at, we have 12 million cells, right? What's the rarest cell type? Obviously, there's a lot of neurons, there's a lot of liver cells, but what's the thing we see the least, right? And so, so the image here on the left is from a disorder called Cartinger syndrome, also known as primary ciliary dyskinesia. And so exactly half of people with this disease will have their internal organs flipped, right? They're a mirror image of the rest of us, right? Exactly half. And the reason for that is that there's, in early development, there's a cilia that rotates one way and sets up your right-left axis, right? And we know about this through the genetics, but if you don't have that, your body just picks randomly. I'm gonna go this way or go that way, right? And so that's why exactly 50-50 end up. And so we can find these cells. They're just 60 out of 12 million, right? That are the cells that have these cilia that are setting up the axis. And they're only there for a few hours. So we wouldn't have seen them if we didn't have this very tight time resolution. We see them appear and then we see them disappear, right? After the axis is presumably set up. All kinds of other things. So, we can look at how cell types give rise to other cell types, right? So we don't know for sure that this cell type is changing into this cell type. But because the frame rate is so high and because we have so many observations, we can essentially put things together that are similar, like lining up frames from a chopped up movie reel or something like that. And for example, decipher aspects about how the kidney develops, right? Or how a nephron develops in the kidney. Different cell types appear, and then kind of trying to reduce these to simpler representations. One challenging piece about the data is that, the way we actually do it is that we lose all spatial coordinates, right? So we get an embryo, we grind it up, and then we look at all the single cells. So we don't know where in the body each cell came from, and so that's a limitation. But there are increasingly ways or technologies that we can use to try and map these back to physical positions. And I won't get into too many details here, other than it appears to be working pretty well. And we can map, cell types that were originally confusing us with respect to where they came from. We can figure out they came from like the lining of the brain in one case, or the lining of the kidney or inside the kidney. And that helps us, figure out what they are and who their neighbors are, right? Okay, and then probably one of the funnest aspects for me here again is time. And so this is just an animation of the emergence of the brain, right? So very early in the window of development that we studied, the brain is, there aren't neurons. It's just kind of this pattern mass. And we can see different kinds of neurons launch from that pattern mass and give rise over time, right? So the animation there is over different time points, and you're seeing things kind of progress like, little rocket ships taking off from a landing pad or something like that. So one of the challenges here has just been that development is just so complex, right? It's here I'm showing, different ways that people have tried to represent the complexity of development, like different kinds of graphs, and there's no really good way to do it, other than that you have to do it. It's too complex for a human to get their head around, a 12 million cell data set. You need to break it down into something that you can kind of understand. And so we've tried to do this essentially by building graphs, right? Or kind of like trees, but trees of not of cells, but of cell types. right? And so the way that we do this, I'm not going to go through this in detail for reasons of time, but in a nutshell, we're finding cell types that look like other cell types. And if they look like each other, then we draw a line between them, right? And if we find another cell type that looks like that one, then we draw another line. And I wish this was all automated. It's big parts of it have been automated by CX, but we spend a lot of time simply looking at this manually and trying to figure out what we're looking at and wrangling the data to make sense. But we've done a lot of this over the last year, kind of during the maybe it's kind of post-pandemic, but it's really come together, and we have sort of at least a V 1.0 of a graph where essentially the top of this graph is a node that corresponds to the fertilized egg, right, essentially. And then as development proceeds, this diversifies or expands in complexity, and the kind of the terminal nodes of the graph correspond to the cell types that you see, in a newborn pup, right? Okay, so in kind of to summarize this, up to this point, right, we have this high frame rate movie of mouse development, right, that we've pieced together in this way. And so one thing we can ask, like, were there any plot twists? Was there anything that we didn't expect to see that we saw that I can tell you about? So I wish there was a better way of showing kind of what I'm looking at here that simplified it more, but I think it's kind of fun to look at the raw data because you can kind of see how subtle it is. So this is a representation of cells of the kidney. Every point here is one cell. And as with that three-dimensional fly around I showed before, we've taken that's 20,000 dimensions. And in this case, we just reduced it to 2 dimensions, right? And so cells that are near each other are similar to one another. And these numbers correspond to different cell types in the kidney that we know what they are, right? Over here, we've taken the same image, and instead of coloring it by cell type, like here, we've colored it by time point. And so you can see the early time points from before the 10th day of development are here. And then as development proceeds, you've got different cell types emerge in these 2 directions, but it's kind of a continuum, right? It progresses very gradually, which is nice and kind of what we expect and allows us to get our heads around the data. But there's one place here where it didn't really make a lot of sense, and that I'll highlight that right here. And so again, kind of the nomenclature here. So E17.5 means 18 3/4 days after fertilization, right? As soon as the mouse is born, we stop using the E and we call it P, right? Postnatal. And so now P0 means immediately after birth, right? And you can kind of see this jump here. Everything before birth is here, and then everything after birth is here. And so that's kind of weird, right? And there's lots of technical artifacts that can cause that. And so we spent a lot of time trying to make sure it wasn't an artifact of the data or something like that. It didn't appear to be. And one of the things that kind of suggested that maybe it wasn't an artifact is that it was happening for other cell types. So if we looked at, not just the kidneys, but the liver, fat, lungs, we saw the same thing, where cells after birth were very different from cells right before birth, right? And this wasn't true for the brain. The brain didn't seem to notice, right, that anything had changed, right? And so again, when we are, I guess the people in Maine collect these mice, no one's sitting there watching for the mouse to be born, right? So they might leave and come back, and then, oh, the mice are born. I'll sacrifice it and freeze it, right? But we don't know how long they were alive. And so Ian went back and did a more careful experiment just to make sure that the result replicated. So here we have a single mouse, pregnant mouse, and with a litter, right? So they have mice of large litters. And Ian was going to do essentially a C-section and take out mice one by one with different amounts of time, right? And let different amounts of time pass, right? But Ian was hungry, and Ian went to lunch. And so when Ian came back from lunch, 3 mice had been born, right? So Ian didn't panic. He said, okay, I'm just going to save those. And he estimated he'd be at lunch for 1 to 2 hours, pretty long lunch, but not in my lab. So again, didn't panic. He just set about collecting the remainder of the litter, right? So basically collecting mice and then allowing them to sort of breathe for no time, 20 minutes, 40 minutes, 60 minutes, or 80 minutes, right? And not only did the result replicate, we could narrow the time frame in which these very rapid changes happen, right? So within an hour, or 40 minutes or an hour, right? But really between about 40 to 60 minutes, we see these massive changes start, right? So here we're plotting all cells from the liver that we see across all of these C-section embryos. And you can see, you know, the 0 looks like the 20 minutes, looks like the 40 minutes, but then, oh my gosh, things start to change at 60 minutes, and then they really change at 80 minutes, right? Same thing in fat cells, right? 0, 20, 40, and then boom, 40 to 60, right in that, you know, as you approach the 1 hour mark, things really shift or move. Same thing in the lungs and airways, right? This kind of shift, right? So, you know, many of you might have guessed what's, you know, part of what could be going on here, right? So the most obvious thing is like one of these things is not like the other, right? So in particular, this mouse has been born, right? So even though we had 6 hour intervals between 2 to 6 hour intervals between every other pair of adjacent time points, the most dramatic things happen 1 hour after birth, and far more dramatic than anything we saw before. But again, the mouse is born, right? And you know, you can this is actually was you know, even though in retrospect, I think it was kind of like, okay, well, that makes sense. It's still something you don't think about very often. But, you know, this moment, right, the moment that you're born is another one of my kids. This transition from the uterus to the extrauterine world is a huge change in circumstances, right? Probably the biggest one you'll ever have in a sense, if you think about it, right? You're just sitting here, right? And then all of a sudden you're out, right? And so there's a lot of physiological things that need to happen very quickly, right? And so, you know, just as an example, right? So number one, you're cut off from mom's blood, right? And so you no longer have access to that source of glucose or sugar. It's really cold, right, in the real world. And then, you know, now maybe it's cold metaphorically, but then it's cold literally, right? And then finally air, right? You got to breathe, right? And you didn't have to breathe before, right? So, you know, this is kind of simplifying it, but you know, they're even in these cell types. So for example, like in the liver, we see genes corresponding to a process called gluconeogenesis kicking on, where the liver essentially is making sugar out of fat, right, as it now needs to, very quickly. In the brown fat, we see a process called uncoupled respiration turning on, where essentially it's kind of like you're burning energy for no reason. You're just shivering, right, for the pure purpose of producing heat, right? Your cells are essentially shivering. And then finally, in the lungs and airways, it makes sense, right? You imagine some transition is necessary. We look at the genes that are changing, and it doesn't make any sense. So this is still a mystery why these particular genes are changing in the lung and airways, but we assume it's something to do with the fact that you're now breathing, even though we don't understand exactly what is going on. And then even beyond these three tissues, there are tons of cell types, including the blood, the kidneys, and others, where we also see these kinds of rapid changes in the immediate aftermath of birth, right? But again, not every cell type, right? The neurons in particular are an exception to all of this. Finally, you know, thanks to Ian's, you know, retrospectively timely lunch break, we can also look at differences in vaginal versus cesarean delivery, right? This was not part of the original plan to look at this, but now we've got a great comparison. Within the same litter, we have 3 mice that were birthed naturally and a bunch of mice that were birthed through C-section. And the remarkable thing is that we also see differences between the natural and the C-section deliveries, but they're not the same differences. It's an entirely different axis of difference, right? And there's a lot of there are a lot of epidemiological studies on differences in outcomes or health for elective C-sections versus natural births that have been attributed to what were thought to be differences in the microbiome of, let's say, the vaginal canal, or kids who are born by C-section versus natural birth. But you could also imagine that there are other kinds of differences that take root through processes like what we're seeing here, right? So, okay, so in summary, you know, there's actually, many of you may have never heard this quote, but there's in the field of developmental biology, there's a very famous quote, which is, it's not birth or marriage or death, but guest relation, which is the most important time in your life, which was this kind of early moment in development, right? But maybe birth is number 2, is my point. Okay, and then of course, as we know, just like in humans, also in mice, development does not end at birth, right? And so our current plans are to continue this experiment and go forward, including, for example, to the moment when mice open their eyes, which is not till 8 or 9 days post-development, post-birth. And maybe at that point, we'll see the neurons start to change because all of a sudden you have a new set of experiences that are coming in through your eyeballs. Okay, so just to wrap up in the last few slides, one thing I want to make clear is that I started out with this example of John Sulston sitting in a chair and drawing out this tree of cells, right? And that is still the goal here, but we have not done that, right? We're taking these snapshots from different mice, we're kind of piecing together what is still arguably a low resolution film. It's like if there were 40 versions of The Godfather, and we're getting little bits of each of them, and we're trying to put together some movie, but the movies are a little bit different, so it doesn't exactly make sense and that kind of thing. So we'd still like to, have one high quality version of The Godfather. And so we're still also thinking about that. And so that gets into this idea of recording, which I'll just touch on briefly. And the idea here is essentially analogous to a flight recorder in a plane, where the flight recorder is part of the plane. It's recording everything that's going on. You don't need it, but if the plane crashes, you've got some sort of memory or record of what happened, right? And analogously, you can imagine trying to build a flight recorder into a mouse, right? And to do it, using DNA as the place that we actually record things, right? So I'm not going to get into the technical details here for reasons of time, but in the last couple of years, these two folks shown here, Janong Choi and Will Chen, have developed a pair of technologies that we think are really powerful for this goal of trying to write into DNA, essentially your memories about what happened along the way, right? And for that to happen during development. So this is literally, but like, the DNA is what you're typing on, and what you're typing is meaningful, right? To something that you're trying to understand, right? And so again, without technical details, we do have worked out a way to do this, where we can write symbols into DNA, and those symbols can have meaning. And what we're hoping to aim for is essentially a symbolic language for longitudinally recording biology inside of cells, right? So and then eventually, of course, you may sacrifice the mouse or that kind of thing, but then essentially you're not looking at the mouse only at the moment that you sacrifice it, but you have some history written into its DNA that you can also piece together, right? And so examples of kind of how we've done this, or what we've done with this so far. So we've done things like writing short text messages into DNA, like Mr. Watson, come here, or What Hath God Wrought, which was Morse's first telegram, or Bound Forever DNA, which is a K-pop lyric. But then, that was just kind of fun proof of concept experiments. And what we're trying to do now is to move towards recording history, right? Recording actual biological history into the genome, into what we call DNA tape, right? And I'll just show you a snapshot of some of the kinds of trees we're starting to be able to generate. So this is like a Sulston lineage tree, but it's larger, right? But it's still much smaller than the tree that we actually would need to capture an entire mouse, right? And the idea is here, though, is that if we can build these kinds of trees, and we can do it for individual mice, right? We, for example, could look at, go back to models like this, right? And look, for example, at I really should have a picture of a normal mouse here that doesn't have this trait, but we could essentially look at what parts are the same and what parts are different in that tree between a mouse that developed normally and a mouse that developed this piebald trait, and ask where the differences are that might underlie this trait. Not only know, how they happened, but when they happened and who they happened to in terms of the tree, right? Okay. And I'll stop there, and thank you all for listening, and happy to take any questions if there are any. So. Yeah.
Did you come across any cell types that were previously well characterized in literature and treated how to about it?
So the question is, like, did we find these cell types that weren't expected, or that weren't? Yeah, so I'll answer that kind of two ways. So one is, one thing we did learn is, I mean, developmental biologists are incredibly good, right? And so whatever knowledge has been built up over the last 50 years is pretty solid, right? And we can kind of match a lot of what we see to the literature such that the overwhelming majority of it is coherent and consistent. And so anyone who tells you there's some magical cell type we've been missing all this time, that's probably not true, right? That being said, I think there are a lot of things that were like poorly characterized and where it was harder to make sense of, at least initially, right? And so probably the best example of that, it sounds like you might have some biology background, but that terms like mesenchyme and fibroblasts, these are like, they're words we use that like, I don't think anyone really knows what they mean exactly. They're kind of catch-alls for a lot of different things, right? Or very broad, loose terms, right? And so within that kind of category, we could see a lot of structure that was not well characterized. So for example, while your body is developing, every organ is essentially surrounded by a layer of this mesenchyme stuff, right? That is not, we don't really understand, or we don't, it hasn't been very well characterized, right? And we were able to map, the subsets that, let's say, coat the liver, coat the gut, coat other organs, and see that they're clearly very different, right? And are probably playing some role in actually shaping the organs that they're surrounding, right? That's one example. Charlie?
Is it a strict tree? It's interesting to see that you have like the spaces of differentiation and the animations looks continuous, but then how do you get to a strict tree? Are there anything that breaks that pattern?
Yeah, so through the approach that we took, we cannot get to a strict tree, right? So through the bulk of what I talked about, we cannot get to a strict tree. With these recording methods that we're developing more recently, or I guess that are maturing more recently, we can get to a strict tree. Like we're not there yet, but it should, in principle, give us a literal tree, because the reality is a literal tree, right? One cell does divide into 2 cells, 4 cells, so on and so forth. Every cell comes from another cell, right? And I guess the other kind of subtle question there was whether we see any discontinuities, like do things leap, I guess you could say.
Like convergence in the actual.
There's both, yeah. So there's mostly it's divergence and continuity, but there are examples where things end up looking the same and you can't tell what direction they came from, right? So for example, neurons look more different early in development than they do later. So they end up all kind of converging, and they're harder to tell apart the longer you go, right? But in many other cases, it's more branching. Does that? Yeah.
So you glossed over the waiting exponential reading cells. Is that because it's a closely guarded secret?
No. Just time and focus. Do you want a 60-second version? Or okay. So it's based on splitting and pooling, right? So for so the way I would describe this is, let's say we were all cells in this room. I don't know if this analogy is going to work. I've never tried it before. So let's say we're all cells in this room, and I just instantly snap my fingers and we all split to 3 corners of the room, right? And everyone who was in that corner got a red book. Everyone who's in that corner got a blue book, green book, right? Everyone came back in the middle of the room. We all ran out again to different corners. And now you got like a red feather, a blue feather, a green feather. Same thing, right? We do it 3 times. 3 to the third is 27. That means there's 27 possible combinations of items you're going to end up with, right? So now imagine instead of 3 corners, we had 384 rooms. And we had 2 million cells that were all getting split into these 384 rooms, right? 384 to the third power is 57 million, right? That means that every one of those 2 million cells is going to end up with a different combination of items, right? Does that make sense? That's essentially what we're doing on a bench. right, with just plates. And the items that you're getting are little DNA tags that we append into the cells, right? That analogy actually did work pretty well. So, okay, I'll use that again. Yeah. We have not had time to look. So, did we just look at, so we tried as hard as we could to alternate. So we did not have the bandwidth to look at a male and female at every time point, and there's no guarantee they'd be the same time point. So rather, we just tried to alternate with the idea that we could figure out how to analyze that kind of data after the fact. But simply because of the number of threads we've been chasing down, we just haven't had a chance to go back and look at it. There are some clear differences. So for example, you can see Sertoli cells in the males and that kind of thing. But getting to more, neurological differences, that kind of thing, I feel like that's a project on its own that we just need to get time to do. So, yeah.
So you said that the change association, with dramatic change, or did it some other metric like the times with it?
I guess if you look at these kinds of and this is it can often be misleading, so you have to be careful when you look at these images. Kind of like alluding to one of the earlier questions, everywhere else in the data set, we saw this continuity. Things were changing very smoothly. And then at this moment, they weren't changing smoothly. They almost seemed to leap, right? And there's other ways we can quantify that leaping in terms of distance and in kind of like distance in transcriptional space, or molecular distance kind of thing. And it's a real thing. They really are changing much more rapidly than earlier moments of development. Again, specifically for that subset of cell types, not brain cells, but that subset, we could see it. We first noticed it visually, just looking at these things like the shrapnel animation, but when we look at it in a more rigorous way, it holds up. Does that answer? Yeah, okay. Cool, well, thank you all today.